In fact, I was not aware Vinod's company is in plastic, more of a productions of the plastic wares required for the regenerative medicine. In fact, I will take little today's speech, considering regenerative medicine as a plastic organ or plastic material. Seeing that journey of today's science going towards the changing scenario of innovation and next corner or next leap of regenerative medicine, next. Revolution of medicine does make sense to understand we are finding some limitations in the current scenario of standard medical care. Next. Now, the next generation of medicine, when I'm talking to you, next slide, next generation of the slide we are talking to you is the science which is going to be a future for all of us. My journey has been by training as a neurologist, as a professor of surgery, and now in last 10 years is been in regenerative medicine. And I have looked this science of journey in a very different way, because now the clinician will not remain individual as a clinician to practice medicine. If you look into future science, there's a definitive basic scientist role in medicine. The basic scientist role in medicine indicates, for the reason, without having basic knowledge of biology, the products of medicine won't be a future for us. The entire conventional medicine will see how it has failed. The entire umbrella of regenerative medicine will see. And in all the stem cells, particularly MSCs do play a major role. How do they play? And rest of the regenerative medicine, along with the stem cells, like if you can see the tissue engineering, organ development, various type of cell lines, the cytokines, chemokines, then exosomes, then chaperones, bioactive proteins, all of them will make a beautiful umbrella under which we can put all the regenerative medicine. The main role is played by the stem cells, that is called the mesenchymal stem cells, though today we started with the mesenchymal stem cells to call stem cells. But let me tell you, in the current 2021, mesenchymal stem cells are no more known to be stem cells within vivo, like within the body. Within the body, they are 24/7 medical stores or organ cells, microorgans, which are secreting more than 800 type of cytokines or drug products, which are taking care of our immunomodulatory and inflammatory and all anti-inflammatory or even regenerative concept of the body. The plastic, by word used is here, does mean that the revolution of plastic has taken place somewhere in 1940s and 50s, 60s. We all know the movie called by name called as Graduate, where the plastic made a big revolution. Next. Now, this revolution of plastic started with the making of everything out of plastic. That is like a regenerative medicine, which I'm going to talk today you. From the small pin to a motor part organs or motor parts. But people thought of making plastic valves and plastic organs. But as we know, when we talk of a regenerative medicine, there has to be some spatial functions in these cells. These spatial functions thus indicate the growth, the multiplication or division, differentiation, and giving signals to maintain the growth. And then again, as we age, the cell lines become depleted and reduced. But the plastic doesn't do that. But that is the difference between plastic and human beings or the regenerative medicine. That is why we failed in making, next you can take the slide. That is why we failed in making it the different organs of the body. But regenerative medicine today has evolved in as a next pillar of medicine, for the reason being, we are looking this as the evolution of basic science, where we started the small molecules of pharmacologicals, antibiotics in 1900, 1940, 1950, where NSAIDs or penicillins and all those were developed during this period. Now, thereafter, we came to large molecules in 90s, where we came to a make of monoclonal antibodies, which were partly biologics, but having target deliveries and target to the tissue, to the cellular parts. But that is also a limitation. And now we are the next age in 2010, where the Science Translation Medicine Volume 5 specifically speaks of pharmaceutical industry long dominated by single molecule, small molecules, now going to a new revolution, the use of microbiomes and human cells as versatility in therapeutic genes or therapeutic applications. This is how the entire change has come next. This is what we look into future. Next slide. This is what we look into future as next slide, the biological plastic or regenerative medicine. Now the story starts with our birth. If you see this slide makes to me a very prominent slide to speak for 3 hours, 4 hours continuously, because it speaks the complete regenerative medicine and its plasticity. In this slide, our journey starts from a single embryo, from a sperm and ovum. The plasticity of the slide, you could understand the regenerative capacity of the, I will remove the word plasticity, but regenerative capacity of this particular slide indicates that when a single cell can make you so plastic, so differentiative, so regenerative, that you can develop a multicellular adult human body, which has got multicellular various organs at every stage from day one of your fusion of two gamete-like cells. One is the ovum and second is in sperm, a single embryo, which differentiates into a small embryo on third or fourth day, what we call morula. Now, these cells are primarily called as embryonic stem cells. Now, here I'll speak of differentiation of types of stem cells. The embryonic stem cells do have a potency of differentiating into all lineages of your body, and not only all lineages. How many lineages do we have in the body? There are 120 types of cells in the body. The body is composed of trillions and trillions of cells. These 120 plus cell lines come from these embryonic stem cells. At this juncture, the body has a totipotency. Totipotency indicates there is totipotency, pluripotency, and multipotency, and unipotency in the potency factor. Now, what is potency? Potency is the potential to differentiate into various lineages. Earlier the embryo, higher the potency, later the embryo, lower the potency. The standard equation pharmacology, this is a standard biological equation which we can use in our daily practice or daily knowledge. Now, when we talk of earlier, then we come to a embryonic stem cell where we call in that totipotency. This totipotency of the cells, which is defined as can differentiate into a complete human being like cloning, because the cell still has not gone into a single expression of gene here. And when it comes to a blastocyst, 5 to 7 days, now these up to 3rd to 4 days morula, if you take these cells separate and put into various uterine cavities for a gestational growth, they can differentiate the entire human being as it is like a cloned one, because it has got an entire regenerative medical capacity of entire human being, including extra embryonic like placenta, cord, organ, biological tissues which connect to the mother, to the uterus, and to the human being. Now, then comes the pluripotency, totipotency, then comes the pluripotency. This pluripotency indicates now the total capacity of extra embryonic tissue making is lost, because it goes into expression of specific CLX molecule gene, which leads to a blastocyst having trophoblast layer outside and inner cell mass inside. Now, this inner cell mass is one which is responsible for you to connect to development of all the organs of the body, that is 220 types of cells, plus the trophoblast carries the potential to differentiate into the lineage of cells of the placenta, cord, and they make the amniotic fluid. What is this important is they make an environment, connectivity, and the feeding of the gestational baby from the mother is the job of this trophoblast. Whereas the inner cell mass gets into the, see the differentiation, total regenerative potential is absolutely plastic, single cell going into different lineages, the plasticity, the regenerative potential. The inner cell mass now carries the potential different 220. Now it starts differentiating into various organs. Before organs, the tissues. Now, when they start differentiating tissues, the potency comes to multipotency. Now, these MSCs belong to multipotency groups. Now, the iPSC cells, what we call induced pluripotent cells, are similar to pluripotency of the embryonic cells, which have been derived in as an biological reversal of aging or biological reversal of cell from a adulcy differentiating character to the embryonic character. This is one which is a challenge, and this was 2012, where the Shinyamanika and Gordon got the Nobel Prize for this work. Then comes to the various organ development from tissue. Here, the tissues has got a very strong potential, what is called as a stability. Every cell has to differentiate to a specific level, and where the growth of that organ has to stop. That is called as a stop effect. This stop effect controls, along with the immune system, the differentiation of the cells to certain level where they maintain the integrity between the cells, differentiation at equal potential level, so as the organ will have a specific function maintained, all the cells at level. And at this level, every organ develops a niche zone. What is a niche zone? Niche zone is where the organs after the birth have to grow from a gram level to higher level of adult size. The niche contains the niche cell, the stem cell, and the niche environment, which contains the differentiation and division by two types. One we called is one by differentiating into similar type of two baby cells of the adult growing cell, that is symmetrical, and the asymmetrical when the cell differentiates into adult cell and maintain the embryonic stem cell pool in the body. And as the baby grows, we are born at like a 2.5 kg. And the wonderful of this regenerative medicine science of stem cells is that this 2.5 kg baby, which is born after 9 months and 1 week, develops into a 60 kg of an adult human being. So the beauty of the regenerative medicine is that entire cell component, entire adult cell component, stem cell component, progenitor cell component, their secretomes in the form of various like exosomes, cytokines, chemokines, bioactive molecules, they play a major role to maintain the regenerative. And next slide, to maintain the regenerative, the entire umbrella, next slide, entire umbrella of stem cells or cells. Now, as I was talking to you, why we are coming to the next generation of medical science, that is the next edge of medicine, which I call is a biological science, a biological medicine, which we have learned in the 11th and 12th science, the cell biology. This cell biology is today's making a medicine which is getting to a treatment what we call as a cure, not as the treatment of signs and symptoms. The total signs and symptoms which we look into conventional medicine are to control the symptoms, control the signs, whereas the tissue in the body goes into a complete auto repair system. That is the reason, if you see at the base of the slide what I have written, we carry our repairing tools in our body. The entire tool system is constantly repairing you, whereas the entire system with the pharmacological or small molecule plays in a role of only treating signs and symptoms. The examples like insulin and nitroglycerin, if you take, which are 50 years and 100 years before before we invented. Still today, they control only signs and symptoms, but body repairs. Next, this repairing system is what we call, this is the same slide, insulin nitroglycerin. Next, the repairing system is called as a entire regenerative medicine. And when we want to think on this regenerative medicine, we have to word disruptive. Disruptive means challenging to the existing science. We have to see how we can prove our science of today's standard medical care going into a new science by challenging the theories of today, which I'll show you subsequently. What are the challenges we are facing in case of regeneration, in case of infertility, in case of autoimmune diseases, in case of cancers, in case of neurodegenerative conditions, where we are failing the standard medical care. So I call it the innovative disruption. This is called a challenging to the existing proven science. Next. Now, when we talk challenging, then we need to be scientifically very strong. Breaking the conventional, this science brings us close to the evidence of God. Next, we go to the next science of natural evolution, which will change the future of the mankind and prove beyond proven science. Next slide. Now, this science, when I call it to be a biological plastic, but it's a regenerative medicine as a new revolution. Next. Now, this science of regenerative medicine comes under a huge umbrella, which job of regeneration, restoring, replacing, and repairing. The molecules which come under this, stem cells play a part of its role, but adult cells, then various type of the scaffolds and growth factors, and most important coming genetic science and tissue engineering, all play a very strong role to make the concept of regenerative medicine in the form of regeneration, restoring the function, replacing, and repair the system. Next. Now, looking at this potential of all the cells in the body, adult cells, or stem cells, or all kind of biological molecules, what is strong to maintain the milieu of the body in a reparative science or regenerative science is mesenchymal stem cells. Once upon a time, we used to call these cells as mesenchymal stem cells. Today, no more we call them mesenchymal stem cells, but we call them the medical stores of our body, multifaceted facilities. They per se in the body remain as pericytes, anchoring along the capillaries. In vivo, they play their all your milieu of reparative environment maintained by MSCs, by these pericytes, when response to the injury, response to the infection, response to the degeneration, they get de-anchored from the capillaries, given milieu of in situ repairing, differentiating the potential of immunomodulation and also immunomodulatory effect they will give. Whereas we call them universal healers for the same reason. We will see some of the classical pattern behavior of MSCs, but no more. But the same MSC, when I take out in a vitro in my tissue culture lab, do you know the wonder of these cells? We can make them dance and differentiate into various lineages. The same cell in the body cannot do that job. Once upon a time, the science I call medicine as a dynamic science. Reason, everything is changing every day. MSCs, once upon a time, were thought to be differentiating in the body. But now, Arnold Kaplan, who is the father of MSCs, has proven that these cells no more hold the status of stem cell in the body, but they hold the status of medical stores or cells which can give the environment of differentiation of other environment healing potential in the body. In the same cell, when we take in vitro, put in a petri dish, we could use various differentiating indicators like mesogenic process, like mesogenic process, where the cells can be danced to differentiate into various lineages like chondrocytes, osteoblast, mesenchymal cells, immunomodulatory cells, the muscle cells, and various lineages like lipocytes, using at different levels the commitment of that cell to differentiate by using various factors, and whether in the bone marrow, whether in the fat, these cells have a potential to differentiate into various lineages when we take them out of the body and go into laboratory. So we make them dance different. They become stem cell when you're outside the body. This is called as a mesogenic process of the stem cell. No more mesenchymal stem cell. It comes from a pericyte in the body. I hope I'm making very clear concept of stem cell, MSc, and regenerative medicine. Next. Now, having known this potential in the body, if you see the MSCs, how do they act as a pericyte becoming MSC and creating a repairing mechanism or repairing environment? Now, this repairing environment, as I told you, pericytes anchor along the capillaries. To the injury, they respond by secreting various kind of secretomes. These secretomes, if you see, are a kind of immunomodulatory molecules. They are molecules for anti-inflammatory effect. They are molecules for effect getting tropic effect. When the environment becomes repairing, these cells go back again and anchor along the capillaries. And the range of secretome, if you see from ILs to TGFs to growth factors to VGF, anything, if you count as a drugstore, they are 24/7 producing in the body and remain as a molecule to have a medical store functioning in the body, producing 800 plus molecules at different, and their secretomes also will change depending on the environment where they are. They could be inflammatory alone. They could be anti-inflammatory with regenerative. The same cell depends on the environment in which the entire cell has to act. This is called as a regenerative medicine, a plasticity of the cells in the body. Next. This was the Chang's hypothesis. If you see the MSC bioactive factors in the body, in the front side, what I'm talking is allo cell therapy within the body. They play a major role of having secretomes. Whereas in the tissue engineering or in the lab, we can play them along to differentiate various lines and go into regenerative of tissue engineering various organs using scaffolds, or turn over the cell lines to maintain the laboratory to use those adult cell lines for therapeutic or drug discovery and all other lab works like drug discovery or into disease models to produce in the lab to check the drugs or their effectivity. All that has a major role to play here. Whereas in the body, bioactive factors and immunopreservation or immune environment to maintain. Next. Now, having various models of these, as I was talking to you, MSCs we can call as now in the body medicinal signaling cells, that is MSC, instead of calling them mesenchymal stem cells. In the environment of the body, a complete regenerative microenvironment. Next. Now, with this idea of regenerative medicine potential of MSC, the entire portfolio which I designate to MSC, drug-like action, multi-modal at a time is immunomodulatory, anti-inflammatory, anti-apoptic, cell differentiation, and a microenvironment enrichment and angiogenesis and neurogenesis. Next, now when we have this potential of cell, now I call this as an biological plastic. One drug, multiple actions, regeneration, leading from a single cell, giving all the actions in the body, not like a micromolecules of pharma products. One product acting on diabetes like insulin products or any kind of anti-diabetic drug, anti-hypertensive drug, all those have single actions, whereas MSC has action which gives an entire regenerative potential. Next, looking at the total application of regenerative medicine of stem cells, 3D printing is one more technology which comes after regenerative medicine, not only looking at MSC or adult cells as a potential. These same in the lab, cells which we differentiate or regenerate or differentiate into adult cells could be using 3D printing technologies. We could develop novel bioactive molecules. We can develop a complete cell sheets. We could develop the multi-layer cells, multi-layer sheets of the cell, which could be used as in the failing organs in the body. We could use them regeneration of the organs in the body, or in the lab we could develop various organs like pinna of the ear, heart, which we have seen at West Forest University. Some of the top researchers in neurosurgery, they are doing organ developments. Next, you can go ahead. Now, decellularization in regenerative medicine is one of the technology where we use the scaffold of the organ as the scaffold to hold the entire non-cellular components after washing away or decellularizing the entire cell components, and use this technology, decellularization technology, to develop various organs by seeding like various lineages of cell in the organ and putting these decellularized organ with the seeded new organs, new cells in the incubators to take up the new function. This one more, the regenerative potential. The details may not have today time to talk on it, but definitely this decellularization has overcome the limitations of 3D printing, where we have challenges of developing tubular organs, tubular parts like capillaries, ducts, or arteries. We can't develop in that, but the solid organs without that can be developed. But in place of solid organ development, I would always insist, looking the regenerative medicine to failing organs, use multi-layer sheets or cell sheets prepared on various scaffolds of alginate or various kind of polyglycol sheets and other sheets, which we can seed the cells and use those for various environmental failing organs in the body. Next. Now, the levels of using, next slide, the levels of using regenerative medicine in human application as a cell, as a biomaterial, only scaffold and cells will lead a support of entire genetic manipulation, growth factors, cytokines, and also develop the mechanical forces. This plays an environmental factor to play to hold the organ development in regenerative medicine. And this is going to change our future for medicine. And the tissue engineered can be done in the lab, which will be using for the therapeutic applications in the aging process or organs failing because of the morbid conditions which come to us because of our lifestyle. Next slide. The application in the lab using regenerative medicine, if you see the cells going into a cell culture, expansion, then going into on various scaffolds and 3D printings, and again using the same material back to the organ development. This concept is a tissue engineering or 3D scope. The next level, next slide. Now, same thing we could use in the eye application. I'm using the applications now, regenerative medicine, in printing process, 3D printing, or any other kind of cell sheets in a dental, middle ear, in bones, heart, lung, pancreas, particular all the tissues of the body. Here I have got two challenging conditions where I would say in the liver and the kidney, where we are organ transplantation, we, if you see as a clinician, has got two important challenges still today. Still today, in spite the organ transplantation has started 50, 60 years before, the two questions are not solved still. The immunological rejection, and we come on immune modulating drugs and steroids to retain the organ. And second is source of the organs. How do we get the donations of the organs for the application of regenerative medicine in the human being? Now here, if you see the central figure of cell sheet can replace the entire organ transplant theory by using these cell sheets, as because the small number of cell sheet, 3 to 4 layers, will not require tubular blood organs which can supply the RBCs and blood cells, but cell-to-cell communication can take place, the nutritional requirement. And once these cell sheets are applied on the failing organs, the liver and kidney, the organ itself acts as a bioreactor, body acts as a bioreactor, and the new capillaries, new ductal systems develop in this new cell sheets by natural environment created there by their cytokines and chemokines. How wonderful regenerative medicine can change our therapeutic applications in future using stem cell technology. Next, the application of this regenerative medicine, particularly in the cartilage sheet, is not today, has been trying for more than 10 years. And now these sheets are clinically available in the market as chondrocyte sheet, maintaining the extracellular matrix, where the weight bearing of the matrix of the cartilage comes by natural mechanical forces which develop in the body during the differentiation and adherence of the sheets to the surface of the chondrocytes, the bone surfaces of the osteocytes in the particularly joints, and take up the same kind of mechanical force which normal cartilage would take it, like layered chondrocyte sheet. Regeneration will take a hyaline cartilage is confirmed now. Next level, next sheet. This was one of my slides which I went to US with Cell Sheet company to bring this technology in India. Next slide. Now, NK cell, again, modulation using the NK cell adult cells, a part of cell technology in immune modulation for the cancer. NK cells are known for targeting the cancer cells or mutating cells or microbe, bacteria, and other foreign body organs. And they could be used as the targeted treatment for cancers, as well as IL-2 activated cells, we call them interleukin-2 activated cells, to differentiate into the potential of having immune modulation in the body, again, anti-cancer effect, as well as antiviral effect. Next. This is again what I have done in Japan for Osaki methods, which we have Cellex Corporation with StemRx. The next will be like CAR T cell technologies, which probably in future, next slide, which we are saying to develop the CAR T cells. Next slide. CAR T cells, also one of the part of regenerative medicine, where the T cells with genetic modification are made targeted to beta cells or lymphocytes, where the malignancies of the beta cell origin like CD19, CD22 on B cells could be targeted in lymphomas, various type of lymphomas and leukemias, whereas now the same CAR Ts are becoming CAR NK cells also in reconstruction. Next slide. Process of genetic manipulation for the cells with CRISPR-Cas technology, CRISPR-Cas9. For it is called as gene editing, which is developed from the very natural process of virus entering the bacteria, keeping the memory, and this memory carried forward to target the next infection. The same way could be used here. The next slide, because CRISPR-Cas itself is going to take a half an hour for me to talk to you. So the old dogmas of medicine are changing with the new regenerative medicine. Next. Now in the brain, we were taught in the medicine that we have a fixed number of neurons. Now The complexity of the situation is that we develop the neurons in the lab. So no more theory. There are very indicative niche areas of the brain where paraventricular areas or subthalamic areas are where we have niche zones of the brain where niche stem cells, the neural stem cells and niche cells are there, which maintain the wear and tear of the brain in case of the neurodegeneration. Next. This system of neuroproliferation, I will not talk much on this embryonic development of the neurons, because neurons are one which are the first cell differential lineage in the body, in the embryo, before the other lineages develop. Because reason being that these neurons, 100 billion body neurons, brain neurons, they develop within 3 months of one, and then they proliferate. Then they migrate to different zones to specialize next, and then they have synaptic connections. The next slide shows how the plasticity or the regenerative medicine plays the entire spurning effect of various axons and neurons will be growing in the first 6 months of life, where the child has the highest effect of neurogenic retention power of memory or learning abilities. And as we grow older, these synaptic connections disappear. Next, the growth curve itself shows the infertility dogma, as I said you, what was taught in the standard medical care, where we use pharmacological molecules or hormones and drugs for treating infertility. Now, no more this dogma start, where we thought initially the egg at puberty are only 3 to 5 million, 5 lakh cells, and at each menstrual cycle they deplete. But no more this is challenge, because subepithelial embryonic progenitor ovarian stem cells are known to exist in the next level. In the ovary, the entire environment of the ovary goes down because the change in the milieu of the hormones, of which are supposed to give signals to differentiate the cells, they don't differentiate. And the fixed pull dogma is no more. Next slide. No more exists, whereas it becomes the dogma. Next slide. It becomes dogma from fixed pull to the stem cell model, and how the ovarian stem cells are isolated can be used for treating infertility. This one slide. Next. All concepts of regenerative medicine, everything. This you. The improvement of the micro environment of the stem cell to function is very important. Now, in the cell compositions, the bone marrow, very important in the adult is VSCL cells, very small embryonic stem cells, which are very important to maintain the regenerative potential after the chemotherapy or adult after going radiotherapy, or in the aging women's, which can give a regenerative potential to the ovaries which have stopped their ovulation, or testes who have stopped their gamete formation, using these VSCL cells from their own autologous regenerative potential from the bone marrow. Next. Now, DNA chip is another technology in regenerative medicine, which is called the tissue nanotransfection. Next, we're using body as an bioreactor. The small DNA chips can be placed, as I have shown you in the blue color chip. Then a small plasmid, like this plasmid is a genomic component, which gives carrying a specific biomaterial, is placed on the DNA chip, and electroporation next is used to target. Next, this is a permission for us to take a clinical trial in India, and the tissue under the skin somatic cell will differentiate into various line of lineages and use these cells for regeneration of various organs. Now, no more we are bypassing with this technology iPSC cells and embryonic stem cells, which has got limitations in clinical applications because of their challenges having little bit going into a malignant character, because we lose the control there. The cells have little challenges of few social and ethical issues, and also having little challenges of malignancy. Whereas this adult to adult differentiation using DNA chip bypasses this and makes our body as a bioreactor next, and use these cells in future evolution of medicine as biological plastics or regenerative medicine. In conclusion, if you see, next slide, before. In conclusion, if you see, you understand that the stem cells of regenerative medicine of 20th century have a greatest significance changing beyond the scientific achievement will not be from the field of space science or computer technology, but for tissue engineering and genetic medicines of regenerative medicine. These are many of my research papers, which I have recently published in the PubMed journal. neurogenic bladder using stem cells, autologous stem cells treatment. Likewise, I have got a lot of research papers published recently. You can just scroll them fast, Kumar, so that we can finish it talk at the earliest. These are some kind of Westlaw. This is my patented technology. This is my approved. This is my approved presentation. Approved clinical trial going in US with Western Institutional Review Board. Then various presentations of my patent. These are my patents. Next, scroll Kumar fast now. I don't think I need. This is a patent. These are various publications of mine, collaborations and technologies which I am working with the by recognitions with various organizations, my certificates of GMP, and my present various publication media has covered my working. The youngest baby in the world I have treated recognition by various authorities. And my dear friends, with these presentation of regenerative medicine in stem cells, I conclude my talk having given you a complete entire spectrum of regenerative medicine, not only limited to stem cells, but beyond hope. As we constantly strive toward the future, we find technology and scientific breakthroughs moving as towards a direction that was never imagined. The ongoing discovery of stem cell treatment has given rise to a new alternative technique of treatment and a new way of control of our lives. Stem cell therapy has emerged as one of the most exciting gifted field in biological science today. Our wellness as we live fuller and healthier life with a new hope. The journey through stem cell treatment of regenerative medicine is exciting as one art would enable us to help live their full of dreams and long healthy life. Thank you for having a patient hearing to me. I hope I have covered that.
Thank you so much. Thank you so much for this interesting session.
Repairing kids in your body are the stem cells. Thank you.
Thank you so much. Thank you so much for this interesting session. Now, I request Vinod, please begin with our next presentation. As I have introduced in the sessions, we have another more sessions by Vinod.
Yes. Hi everyone. Very good morning once again. Thank you so much, Dr. Mahajan. It was really delightful to watch the session, and it was so informative for in the terms of how the regenerative medicines are moving, and your research was really valuable to understand a lot of aspect altogether into it. So here I would like to introduce Abdos lab technology to all our attendees, and how we are making a great change in the terms of stem cell research. I would say in the terms of cell culture technology that our product line are really helping in forefront currently. And here I'm having a small presentation for you all to talk about that. And here we go. So if my slides are visible, so I can get a quick yes from Jiten.
Yeah, it's visible, Vinod. You can go ahead.
Okay. So I would like to give a little bit introduction about Abdos lab technology, how we are, as I said, how we are making a difference in the terms of plastic consumable range for all the leading research in life science space. So a quick snapshot, how we established and how long we came so far. So Abdos lab technology started its business in the terms of plastic ware consumables, and which range having from liquid handling to we are having the complete range up to equipments, and where we are having a lot of product line, and today we have collectively we have created more than 1,800 product line in the portfolio. So we started our journey, dated back in 2009 with just started with 3 product line and few handful members, and by the time in the year 2010 and 11 we almost had introduced 40 new product line, and it became the few product line became one of the very fast-selling product line for us. That is Abdos centrifuge tube, which is really captured the Indian market by the time. And at the same time, people who are really working in the stem cell and the cell culture. The few critical application which also solved by the centrifuge tube technology product line as well. So then we moved into 2011 and 12, where we added more than 17 new product line, and we opened our global market. and Abdos became one of the few manufacturers from India, and which is driven by the Made in India in the global map. And we became one of the fastest moving product line in Europe, in USA, and so on with the Asia region as well. In 2015, 13 to 15, we made our century in the terms of product development, new product addition, and we collectively added 130 product line. And at the same year, we launched the cryoware and microbiology product range. So cryoware range became one of the very integral part of our planning, because most of our researchers across the globe really asked for this product line to be introduced in, and the focus was to help all the researchers across the globe with a very affordable product line, at the same time creating a very quality standard portfolio for these product range. So cryowire is again now being used for the stem cell and cell culture technology line product too. Moving to 2015 and 17, we added more than 188 products line, and one of the reversible product line I would say is Abdos super filter tips, which is became one of the key benchmark for us. And today in the times of COVID era, super filter tip really helped all our researchers, and I would say that pathologists who are really working in the diagnostic centers, and this particular product line is helping in the terms of cross contamination and the stability of microRNA. So it is this particular product line is not only restricted for with the diagnostic. I would say it's moreover into the microRNA research, and at the same time it's really helping in stem cell culture, cell culture, because filter tip is one of the very key integral part of the entire ecosystem in cell culture analysis, and I would say the work which is surrounded with it. And in 2017 and 19, it was, I would say, one of the beautiful year for us, and we added more than 247 product line, and launch of Abdos European subsidiaries started dated back in 18, I would say, in Venlo, Netherlands, and that's how we started helping and supporting our European life science researchers and the scientist community there. So I would say this is one of the benchmark where we have represented our brand in University of Oxford, and very big institutions who are really working in terms of medicine and I would say vaccine manufacturing. Right now, it's become very quite vocal with the collaboration of Serum Institute. So, but our support for these institutions were dated back, which is starting in 2017. Now, moving to the next slide, again, year 2021. It is we have created 10 years in the life science space, and the product line we are right now we are offering, it is 400 plus, and this is something I would say it's having a very vast portfolio. However, I'm going to talk about it, and this year particularly is I would say one of the breakthrough, and I would say that is one of the milestone for us in the terms of the supply. We have supplied more than 400 filter tips. So that is something I would say huge responsibility as a manufacturer, or I would say as a company, because the way COVID cases has increased by the time, and this particular product line has helped the diagnostic network across the globe. So we also featured in leading research journal by the time, and at the same time, many news agency has recognized our efforts to support the research in the tough times. So StemRx became one of the integral part for the VTM application in COVID-19 sample collection, and at the same year, we also achieved IVD certification for our major product line. And this is something I would say a few benchmark which really required, because in stem cell culture technology, IVD is always one of the main certification that should be validated with the product line, because the product is really effective and efficient and having very critical domain at the same site. So I would say IVD is something we achieved, and right now it's helping our researchers, our customers, and I would say this is something one kind of achievement for us. At the same year, we also achieved our forensic DNA grade for almost for all the product range, which is significantly contributing in forensic applications and crime. I would say more of the crime scenes technology, which is really relying on the forensic DNA grade product line, which is something we are offering, and we have started this particular portfolio for across the globe as well. So this, I would say, this is one of the another benchmark in the terms of standard of the quality and the quality validation from the research end as well. So by the time you had a huge demand over the microtip and liquid handling portfolio, so adding 1000 XL and 1250 microliter pipette tip, which is again being used in cell culture as well, because they are demanding a very good length pipette tips, and that's how this particular portfolio is helping them. And the launch of cell and tissue culture product range became one of the very effective landmark for us, and that's how we are serving almost more than 10,000 customers globally in with cell and tissue culture product line, which is stem cell is again one of the portfolio for us. So just a quick view about how we are moving ahead and what are our 5 pillars. So yes, we are one of the largest manufacturer for plasticware in Asia, and we are having our manufacturing plant in Roorkee, which is having capacities to manufacture more than 1 billion plus centuries tube and other associated product line as well. So it is, I would say, we are fully equipped to handle such product line and help the researchers across the globe. Yes, so as I said, the 400 plus product line, which gives ample opportunity to researchers to explore the product line in terms of whatever the work they are doing. So having a complete ecosystem is always a very advantage for I would say for everybody. So the moment we have launched more than 400 plus product lines, so it really gives a very good outlook to our customer, because they can have a very good hands-on experience with those product line, and at the same time on one roof they can explore these product line for their research. Coming down our coverage, so it is more than we are having coverage more than 65 countries plus. So we are having more than 10,000 plus customers globally. That is completely, I would say, in last 10 years, this particular benchmark has really uplifted our confidence that yes, our product line is really making a difference across the research front, at the same time in the terms of diagnostic as well. So this particular testimony, I would say, that is something is going keep us going, and we are keep adding the new product line to help our community at the same time. So this particular, I would say, infrastructure in the terms of product, in the terms of customer, and the global coverage is quietly associated with the kind of validation we are having. So we are having more than 21 quality certification in the house. So it is something, I would say, which talks about the product quality, which talk about its authenticity. I would say that has become one of the very integral part for us, and it is one of the pillar in Abdos Life Science. Coming down to the next, so the moment we say that yes, we are having the lot of certification into place. So these certifications are already validated by the leading research journals. So Abdos product line are the part of the citation program with the leading research journals. So every year we get an ample number of citation across the globe. So here my next slide is talk about that, how our research journals are moving and how people are really loving these product lines. So the few research journals that I would like to highlight here is Springer, Royal Society of Chemistry, Science Direct, and Bioprotocol, and many are in the same portfolio that really validating us. And I would say in the very critical domain they are working. So I'm having a few slides which is talked about and how our product line is being used in the same domain. So here you can see one slide on my screen that it is talks about our centuries tube, which is used by Bhava Atomic Research Center in India, where and this particular article was cited in Royal Society of Chemistry. So likewise, there are numerous links, which is where I would say this product line is being used, validated, at the same time cited by the research community. So yes, I would say that is something it gives a very good confidence to us. Now moving to the next slide, I would like to introduce you with the product portfolio what we are having. So here the first portfolio we are having is centrifuge technique. So here in this portfolio we are having micro centrifuge tube, Rex, and I would say other product line, which is very fruitful for the initial research, which is, I would say, very incoming product line for every single research, which is starting from the very basic point. So however, particular product line is centrifuge tube, which is also used for cell culture media preparation, and I would say the liquid handling. So Abdos centrifuge tube, which comes with varied range in 50 ml and many more. So this is one of the biggest portfolio for us. Moving to the next, so another one is the Cryo Technique, where we are having cryo vials, storage vials, cryo boxes, and more. This is one of the very integral part of the cell culture product line as well, and it is also effectively managing for in the terms of vaccine manufacturing, vaccine research. So because it's the temperature dependent, and our cryo vials are also featured in leading research journals. So this is again the one portfolio that is really helpful in the terms of the cell culture research. I would say in the extension of cell culture, stem cell technology, and regenerative medicines. Another one, which is liquid handling, and this is, I would say, the backbone of every research, because without tips and pipettes, of our research always it doesn't take that that empowerment, because transferring a liquid is always a challenging job. And our particular portfolio is having more than 43 category of pipette tips, which is from standard pipette tips to low retention. And then we are having the filter tip technology. So low retention and filter tip is, I would say, one of the very key product line which really help in this domain, because whenever we are running out with your precious samples, and you can't go with any standard tip, right? So low retention is something I would say is really helpful in this aspect, and which is also equipment, I would say, added by and associated by filter tip technology, where you don't want any contamination in your regions, or you are really, really afraid of that. Okay, maybe any foreign particle going to make a big problem with your cell culture or either with your sample. So this particular product line is really, really helpful in I would say, in the terms of liquid handling across the ecosystem of the research. So coming down to the next slide, so this is the another range. We are having the microbiology, where we are having packages from 60, 90, and 140 mm, where we are having the soft inoculation loops as well. So this product line is also really helpful in the terms of the basic research of microbiology. And now I'm having very big news for all the attendees, that we have started the first ever robotics manufacturing of packages, where our complete manufacturing is based on the automation. So this is one of the landmark for us in the terms of the expectation from the people, that they are looking for more of a robotics enabled product line, and the packages which is falls in that category right now. So this is one of the area that we are really, really helpful for microbiology, and at the same time it's also helpful in the cell culture. Another one is the biggest portfolio, which is which may sound quite relevant today based on the topic, webinar topic. So cell culture is one of the product category we are having, and here we are having cell culture flasks, tissue culture treated plates, cell culture dishes, bioreaction tubes, and roller bottle, and many more. So here you can a lot of information about it through our website, which is having the complete details about this portfolio, so you can have a look into it. Another one is the PCR technology, where we are having PCR tubes, plates, and PCR strips, so which is again one of the portfolio that in the molecular biology, in genomics, which is helping other researchers. And this particular portfolio is really, really helpful in the terms of, I would say, COVID-19 diagnostic and the testing algorithm, because right now we are in the forefront with this product line, which is helping in the current scenario. So apart from these portfolio, we are having electrophoresis system. We are another supported by the multi-purpose racks and boxes, and the bottle and carboys. So these are the product line, again, we in the pursuit, or I would say in the terms of ecosystem that we are developing. So everybody can get, any researchers who are going for a particular research point of view, so they can get all the product line in one roof. So which is also joined by, I would say, the general laboratory product line, where we are in beakers, cylinders, and many more. So this particular portfolio has 400 particular plus products, and I would say this is something that we are always ready to offer you in terms of that. So a few quick glimpse I would like to highlight about our portfolio, that is the cell culture, which is I would say quite relevant as for today's topic. So here we are having these product line, which is, I would say, going to make a significant contribution in the terms of your research as well. So as I said, cell culture flask, we are having tissue culture treated plates, cell culture dishes, cell scrappers, cell strainers, and roller bottles, bioreaction tubes, and also can buy the media bottles. And another product line that we have launched this year is Biofuel bottle filtration system, which is quite, I would say, one of the impressive product line, which is helping in the terms of filtration of these research. And this is something right now, initially we have started this particular portfolio, and I hope this is something from this particular line is going to help you in the research. So at the same time, few product line, I would say MCT and centrifuge tube, which is again help in the terms of the same line, and which is really helpful. And it is also, we are having the extension of the cryo vials, where you can have a look into it, and the complete information is available on our website. So here I'm not going to make a lot of I'm not going to consume a lot of time of yours, so I'm going to end my slides here. So I hope you get a little bit understanding about what about the Abdos and how Abdos is working in the terms of life science field. So this particular information is going to help you in the terms of your product choices, and of course is going to make quite contribution from our side to help you in your research.
Thank you so much. Thanks, Vinod, for this amazing product insights. So let's move towards like Q&A sessions. So Dr. Mahajan sir, we have one question for you. The question is asked by Mr. Olivier Mustangi from Rwanda. He's asking, sir, thanks, Dr. Mahajan, for your presentation. Remember my son Manesi from Rwanda in Africa has 13 actually with autism and follow your protocol of stem cell 3 years ago at StemRx with valuable improvement with no more hyperactivity only, but uh, language and physio not really improved so far, not expected outcome after stem cell process. My question is how many times the sandwich protocol can be applied to have visible results, and what is the best age to have the quickest results? Oliver from Rwanda. Sir, please unmute you, sir. Unmute. Your voice is not coming.
Mr. Olio, thank you for participating in the seminar, webinar, and having a complete hearing to it. Your question about your son, Manasseh, any one cycle of sandwich protocol leads to a certain level of improvement. And the child Manasseh has both the components. One was the spasticity because of the hypoxic injury, and second is the autistic behavior. Now here, the part of the treatment we have got for the spasticity will require another one cycle, and thereafter some injections which I will prefer to give him intramuscular, which will give him release in the spasticity. I look forward, Manasseh, to have by another one treatment saying visible changes in the spasticity and physical changes, which will make him ADL, what we call the activities for daily living, to reach to the life where he can manage his own life. Maybe one more cycle he will require. Thank you.
Thank you so much, sir, for this answer. Hope, Oliver, you're satisfied with the answer. If you have any questions or clarification regarding Abdos insight or any question regarding today's session, you can simply put it here on Q&A tab. We'll convey a question with the speaker.
Yeah, Dr. Jitin, can we have a quick Q&A from the who are right now attending it, and if they are willing to ask more questions, so we can give an opportunity to them. So quickly, Dr. Mahajan he's here to answer those questions, so we can quickly take those questions if anybody is willing to ask now. I guess yes, Jitin, are you there? Yes. So I think
if you have any questions or clarification regarding any of your any of today's presentation, you can simply mail to Vinod. Can you have your email ID so that I can broadcast your email here so everyone can get it?
So what I'm doing is I am just sharing my email address to
You
everybody.
only broadcast there only. You only broadcast there.
Yeah. So anybody is having any questions, so what we can do is we can take your questions on the email ID which I've just shared. Okay, and here what we'll do is we'll transfer your questions to Dr. Mahajan, and he can effectively answer whenever he's getting time. Okay, so I hope this particular session you guys have enjoyed pretty well, and Dr. Mahajan was pretty impressive with the content he has delivered. It is really insightful in terms of how the regenerative medicine is moving. And I would say, we who are here today in this session, so they are researcher by heart. So everybody, love to hear what is going on in terms of how the medicine market is moving, how the new technology coming in, and at the same time how these technologies are functionally viable. So thank you so much, Dr. Mahajan, for delivering this session, and we really liked it. And I hope, like me, and who are joining today here as attendee, they have also got a lot of insight from this session.
Thank you so much. Thanks, Vinod. Thank you so much, Mahajan sir, for joining us today.
Everyone.
And thank you all attendees for stay tuned here with such a beautiful number. I am really like, and please stay tuned with me for next coming session of Microbios India and Abdos Labtech. So I think it's good to go, Vinod.
Yeah. Yeah. So here we are now. We would like to end this session right here, and we'll see you in the upcoming session. Bye bye. Take care.
Thank you so much, all. Bye bye. Have a good day. Have a good weekend.